Let's jump right into this explainer and set the scene. Imagine you're looking at a single drop of lake or ocean water. To us, sure, it's just a tiny drop. But down at the microscopic level, it is literally a monster movie playing out every single second of every day. In these vast aquatic environments, nutrients aren't evenly distributed. They fluctuate wildly. And, well, when food gets scarce, survival demands some absolutely astonishing and honestly pretty brutal evolutionary adaptations.
Which brings us to a brilliant quote from the researchers behind today's source material. They describe this process as fly fishing for flagella. I mean, it sounds like some bizarre microscopic sport, right? But it's actually an action-packed hunting strategy used by a specific type of filamentous bacterium called Aureispira. We're going to break down exactly how this tiny predator fishes for its food.
To really understand this microscopic apex predator, we first need to define its primary method. The technical term is ixotrophy. Basically, that translates a really dense scientific concept into a visceral, everyday image: bacterial flypaper. It's a contact-dependent predatory strategy. So instead of just passively absorbing nutrients that happen to float by, these filamentous bacteria actively catch, stick, and bind living prey cells directly to their own surface. And then, they completely lyse them, meaning they violently break them open to consume whatever is inside.
Let's look a little closer at how this battlefield actually operates in Section 1, Microscopic Hunters of the Deep.
We are looking at an environment where intense nutrient scarcity forces bacteria to evolve, quite frankly, sci-fi-esque weaponry just to survive. This all builds into an incredibly coordinated, two-step assassination process that researchers recently uncovered using advanced imaging techniques like cryo-electron microscopy. The hunt always follows two distinct phases. Step 1 is the catch. If the predator is on a solid surface, it creeps up using gliding motility. But in liquid, they use molecular grappling hooks. Then comes Step 2, the kill, which is always a contact-dependent puncture.
Let's zoom in on that incredible first phase in Section 2, the grappling hook reeling in prey.
When freely floating around in liquid, Aureispira doesn't just passively bump into its prey and cross its fingers. No, it actively throws out extracellular rods equipped with literal terminal grappling hooks. Researchers actually watched this in action against prey like Vibrio bacteria. Aureispira uses these hooks to lasso the swimming prey by snagging their flagella. You know, those spinning, tail-like appendages the prey uses to swim. It is a highly targeted, physical capture, reeling the prey in entirely against its will.
Once that prey is reeled in and securely stuck, the predator transitions seamlessly from catching to killing. That brings us to Section 3, the lethal spear, puncturing the prey.
This brilliantly illustrates the two totally distinct biological weapons that make ixotrophy possible. First, we have the catching mechanism, the type 9 secretion system, or T9SS for short. These are those extracellular heptamer grappling hooks acting as the lasso. Then we have the killing mechanism, the type 6 secretion system, or T6SS. This is a cell envelope anchored contractile injection system. Or to put it in much simpler terms, it functions exactly like a spring-loaded molecular syringe. Basically, it's a spear designed specifically to stab the prey once it's been caught.
So how exactly does this molecular spear work? Well, the fatal blow is highly mechanical. First, a tiny antenna on the T6S weapon detects that the prey is in close direct contact. Once triggered, this elaborate trans-envelope complex contracts violently. That rapid contraction expels a rigid inner tube straight out of the predator, physically puncturing the outer wall of the prey cell. Once the defenses are breached, the predator can just consume the prey's internal substrates. And the coolest part? Researchers didn't just guess this sequence. By using single-cell Raman microspectroscopy with isotope-labeled prey, they could physically trace the transfer of nutrients. They literally proved the attacker is siphoning out the prey's insides.
Now, that brings us to a really fascinating genetic mechanism. Let's look at Section 4, starvation switch, toggling ixotrophy. The crucial point here is this brilliant binary toggle. These bacteria utilize an evolutionary genetic switch to survive. See, when the environment is nutrient-rich and food is plentiful, tiny pieces of DNA called insertion sequences, or IS, physically insert themselves into the genes responsible for ixotrophy. This effectively turns the weapons off, which saves the cell a massive amount of precious energy. But when the environment shifts to being nutrient-limiting, when starvation starts setting in, these insertion sequences automatically excise or just remove themselves right out of the genes. That excision instantly reactivates the hunting machinery. It's a completely automated, incredibly efficient survival response.
So we've seen how they kill, and we've seen how they conserve energy. But asking why cells use ixotrophy forces us to zoom way out from the single-cell battle. Why does this complex predatory behavior exist in the first place? And maybe more importantly, what does it actually mean for the ocean at large?
To answer that, let's head into our final section, Section 5, Shaping the World, Ecological Impacts. Because this tiny, seemingly isolated interaction actually sends massive ripples through aquatic ecosystems.
When we really look at the broader ecological impact, we realize that ixotrophy is, fundamentally, a massive recycling program. Think about it. A huge amount of nutrients in the ocean are locked away inside living microbes. Aureispira and other ixotrophic bacteria are out there unlocking those nutrients, making them available to the wider food web. This whole process is absolutely essential for balancing highly heterogeneous aquatic environments. In fact, by analyzing marine metagenomic time series data, researchers found that the population levels of these predators and their prey are deeply connected in coupled cycles. This means these microscopic hunters aren't just surviving, they are actively driving the population dynamics of vast coastal plankton communities.
It is truly incredible to think that every single drop of ocean water contains a hidden, microscopic battlefield, and that the outcome of these battles ultimately balances our global aquatic ecosystem. Next time you look out at the ocean, remember that invisible, action-packed world of molecular grappling hooks and spring-loaded spears that are ensuring the survival of the deep. It leaves us with a thrilling and honestly somewhat intimidating question to end on. With all of our modern technological advancements, are we still only just scratching the surface of microbial warfare?